Humans have blood, a heart, a muscular pump which squeezes this blood and distributes it throughout the body. And then they have a system of vessels. This blood first passes through these large arteries, then into smaller arteries known as arterioles, then into capillaries, then into small veins known as venules, then into the veins which return blood to the heart. There are discrete layers of blood vessels, an outer tunica externa with collagen, which anchors it to surrounding tissues, a tunica media, which possesses smooth muscle, a tunica interna, which has a lining of simple squamous epithelial cells known as endothelia. Um, and so we are familiar with the cardiovascular system in these blood vessels in humans. If we were to ask, what do blood vessels look like in other organisms? How did blood vessels evolve? Here you can see those simple squamous endothelial cells. These are united by junctions. There is a continuous basement membrane around them so they can control movement, etc. Here, this is a little more difficult. First, the first animals didn't have a cardiovascular system, and there were no blood vessels. So sponges don't. Cnidarians, such as corals, jellyfish, they don't. See this hydra, there are no blood vessels here. There are only two layers, an outer epidermis and inner gastrodermis. There are no blood vessels. Even then, when we get into many bilaterally symmetrical animals, such as this flatworm, there is no heart, and there are no blood vessels. In fact, because there are no blood vessels, if you look at the intestines, you can see how much they branch, because the intestines then are taking over the job of the cardiovascular system. So it is the intestines which is distributing material throughout the body. If you ask, how could an animal live without blood or the ability to circulate materials throughout its body? The answer is that this flatworm is flat, hence the name. And if you are flat, a very large percentage of your cells are in contact with the environment, and thus you can have materials diffusing in like oxygen, you can have waste leaving, etc. When we look at some of the early animals of the Ediacaran period before the Cambrian, some of them can be 3 feet long, even up to 6 feet long, but about as thick as cardboard. So once again, this idea of being flat apparently was an advantage. If simple animals lacked blood vessels, then being flat would be perhaps a way around it. So then the question, where did blood vessels come from? Because the first animals certainly, to have blood vessels, certainly did not have anything like the circuits which we find in humans. Well, there are actually 2 types of circulatory systems that we see in vertebrates. And then this will even relate to the chordate ancestors of vertebrates as well. So here with fruit flies, you see there is a heart, and hearts can then pump a fluid into blood vessels. But notice that the blood vessels end, all right? And so that it isn't a closed circulatory system where the fluid pumped by the blood stays in these vessels the way we see here. These blood cells, these red blood cells, they never leave the blood vessels. They just go around and around. So when the human heart pumps, the blood goes around and around, and it stays in the blood vessel. Here in Drosophila, the fluid that was in the vessel leaves and then just goes into the body cavity, the coelom. And therefore, we don't like to call this blood. Instead, we call it hemolymph, because it then mixes with the coelomic fluid, and then this hemolymph can then come back into the heart. And so that's a very different type of system, an open circulatory system. And then the vessel lining would then differ. When we look at other other invertebrates, they can have blood vessels which are more elaborate, and they can have closed circulatory systems. And so here in this earthworm, blood is pumped by these contractile blood vessels. Here, these contractile hearts, and then blood can go, in one longitudinal vessel the length of the body, and then come back um, the other way. Some worms have just very simple circulatory systems. They might just have, one blood vessel that goes one way, and it's linked to the other blood vessel that comes the other way, and that's the circulatory system. And so in invertebrates, we see these. We see both open and circulatory systems, open and closed circulatory systems, and we see the absence of circulatory systems. Now, in the old days, and I remember because I'm old, the way that invertebrates were classified was that um, flatworms, which don't have a circulatory system, nematodes, which don't have a circulatory system, they were classified anatomically as being, a lower set of animals. And when you look at the higher animals, the coelomates, the protostomes and the deuterostomes, these then typically possessed circulatory systems. And so the model was relatively easy, that circulatory systems evolved, say the simplistic forms of nembrutine worms might provide an example. However, since then, genetic evidence has resulted in a reorganization of this. And this certainly has great genetic support behind it, but now the anatomical features, they don't line up as well. So now if you look at these branches, you'll see groups that have circulatory systems, like annelids and mollusks, along with groups that don't. You'll see a group that has a circulatory system, like arthropods, along with nematodes that don't, raising the question. The ancestors of these higher animals, did they have blood vessels? What were the blood vessels like? So currently confusing. Once again, the new classification system is based primarily on genetic evidence, and that's caused kind of a difficulty in explaining how the anatomical features match. One other aspect, as we start looking at the cousins of the vertebrates, what kind of circulatory systems do they do? Well, most of them have hearts. We could argue about whether there is a ventral blood vessel, which is the homologue of the heart, or not in the cephalochordates. There are vessels, but to some degree they're open in that the blood vessel linings don't have good junctions between the cells, so fluid can leave, pass through connective tissue, come back. So not an open circulatory system like you would see in a fly, but then still with some of the features of an open circulatory system here. As a result, these organisms, their blood is a much higher percentage of their body weight. I think the numbers that I read were, say, 40% of the body weight is the blood. You go to the most primitive fish, the hagfish, now you're at 16. You go to the jawed fish, you're about 5. And so circulatory systems have changed over time. The junctions lining the blood vessels, the basement membranes, they've gotten more secure. You don't really have that here, in the jawless fish, more than the chordates, but not as much as the jawed fish. In fact, some have stated that when we consider the blood vessels of, say, the jawless fish and even the jawed fish, they remind us a bit more of the lymphatic vessels of mammals, less like blood vessels. So, the point to begin with is that animals don't need circulatory systems, and the early animals lacked them. Once circulatory systems evolved, there were different kinds, open versus closed, and the vessels in them have changed over time. So, the cells and the junctions between the cells, the basement membrane around the cells, that has certainly changed. By the time we get to the most primitive fish, the hagfish, Here is an image of the blood vessels in a hagfish. Here you can see the dorsal aorta and then the smaller blood vessels as well. Certainly you would say, oh, here we see, arteries, we see veins. When you look in the lamprey, look, there's the dorsal aorta. There are cardinal veins, just like we find in other fish. And so, you would think, here we are, with things that I understand. Obviously, we'll talk about sharks presently. Yes, but there are differences. The blood vessels of jawless fish are more primitive. One of the things I'd like to just introduce is the idea of a sinus. A sinus is a vein that is larger, wider than normal, and with a thinner wall than normal. So you can see here is the inferior vena cava in a salamander, but look at this big wide area here. That's just bigger than veins tend to run. And so it has the name of a sinus. Humans, we have a coronary sinus in the back of the heart. We have dural sinuses in our brain. So sinuses are these wide veins with thinner walls. Well, these were more the norm. When we look at lampreys and hagfish, we see a lot of this. And so before, the arteries and the veins of the higher vertebrates, there were these big, larger chambers called sinuses, which would collect venous blood and slowly return it to the heart, which is why when you look at, say, a hagfish, 16% of its blood, if its body weight is blood, that's larger than what you would see in, say, jawed fish. Once again, because you have all of these sinuses, which are holding this. Hagfish, they are unique in many ways when we consider their blood vessels and their circulatory system. They have more blood volume than any other vertebrate. They have the lowest blood pressure. They have, those sinuses. In addition to a heart, in their veins, they have areas which can squeeze or be squeezed by skeletal muscles around them and help to propel blood. So in addition to the heart, which is providing blood pressure for circulation, you can refer to accessory hearts, where this area has skeletal muscle squeezing to add a pressure as well. As a result, hagfish, they're not very active. They have the lowest metabolic rates among the craniates. And so hagfish are certainly unique. Once again, the circulatory systems which we see in humans doesn't simply, leap into place. And as I had mentioned, we see more similarities between lymphatic vessels in mammals and these blood vessel linings in jawless fish than to the blood vessel linings in mammals. So leaving that, let's kind of jump into systems of blood vessels, and start talking about how blood is circulated to specific parts of the body. Perhaps the most important set of vessels in the chordates and the jawless fish are these. And so, this is a good place to start. So there is a heart. And if we were to start with, say, a jawless fish, remember there are 4 chambers: a sinus venosus, a single atrium, a single ventricle, and the truncus arteriosus, at least in lampreys. In, say, something like primitive chordate, like Amphioxus, this would just be a contractile blood vessel. So this structure, heart or contractile blood vessel, pumps blood through a ventral vessel called the ventral aorta. This has deoxygenated blood. This would then pump blood to the gill arches, where the gills are. There are capillaries which would perform gas exchange. From the capillaries, then we would have arteries drain these areas with now oxygenated blood into a dorsal aorta. So the heart pumps deoxygenated blood to the gills. And then from the gills, then oxygenated blood goes to a dorsal aorta that gets then circulated throughout the body. The number of these aortic arches, these gill arches, the chordate amphioxus has a very large number. The jawed vertebrates all have 6. The jawless vertebrates have more than 6. And so there seems to have been a reduction over time from the chordates to the jawless fish to the jawed fish, where even in human embryos, we have these 6 aortic arches. All right, so this is a very important this is a very important pattern for the vertebrates, because this then links, this links the circulatory systems to the respiratory system. So here we see a primitive chordate from the fossil record, similar to the lancelet alive today. Here you see those gill arches in the lancelet. Notice far more than 6. Once again, by the time we get to the by the time we're getting to the jawed vertebrates, this number gets reduced to 6. Gas exchange occurs in the gills, so deoxygenated blood reaches these capillaries, but then oxygen is removed from water, so the blue deoxygenated blood, which comes to the gills, leaves as red oxygenated blood. And so if you were to look at then a lamprey, here you can see that on the ventral side of the body, there is a ventral aorta. So here's the heart pumping blood into a ventral aorta, which is going towards the head to the left. And then there are these aortic arches, which have afferent branchial arteries carrying blue deoxygenated blood. And then there are capillaries where gas exchanges occurred. Then efferent branchial arteries then bring oxygenated blood to the dorsal aorta. Now, the ventral aorta, the dorsal aorta, these are obviously arteries. There are also veins in the head, which aren't, related to this gas exchange of the gills, like this anterior cardinal vein. So when we look at the blood vessels in lampreys, this primordial pattern inherited from the chordates is important. Note that the blood is deoxygenated until it reaches the gills. In our hearts, the blood leaving the left ventricle is oxygenated, but in a fish that can't be the case, because you go from the ventricle to the gills. You're not oxygenated before you get to the gills. And then we would see the same thing in sharks. But one of the things that begins to happen, and obviously when we get to mammals and the amniotes to even more extreme degree, you can modify some of these aortic arches. So all vertebrate embryos start off with these 6, but then they can be modified. So in a shark, the first aortic arch is modified and reaches, it is evolved around the spiracle. And so we will see blood vessels, and as we'll be getting to later, a lot of the blood vessels that we're familiar with have an interesting history. So going back to sharks, you can see an external carotid, which in my body brings oxygenated blood to the jaw muscles and the head, and an internal carotid, which brings oxygen to the brain, and they come off of a common carotid. But while sharks have the external and internal carotids, they don't yet have the common. That will be developed later, as you'll see. And so once again, this is the primordial pattern that vertebrate embryos start with, but it can be modified. So if you were to ask how are sharks getting blood to their brain and back. And so they have blood coming into the sinus venosus, into the single atrium, into the single ventricle, out then the conus arteriosus here. Okay, and then we could then trace the pathway. So sinus venosus to the atrium, to the conus arteriosus, to the ventral aorta. All right, here's the ventral aorta in the shark. This then reaches, so you can see the ventral aorta coming out of the heart. So there's the ventricle, the conus arteriosus, here's the ventral aorta, and you're seeing that it's sending off branches to the gill arches. These are the afferent arteries, which would then have deoxygenated blood. Gas exchange occurs in the gills as the blue blood then receives oxygen and is converted to red oxygenated blood. And come. And so then you have arteries, efferent arteries, coming from the gills, then fusing to form this dorsal aorta. All right. However, the dorsal aorta is going to go more posteriorly. The internal carotid artery is what is going to take oxygenated blood then towards the head, where there are brain capillaries. All right. And then there is a sinus. So remember that sinuses were once more common. They will be reduced in the vertebrate groups, but there's a thin, walled, wide area that collects deoxygenated blood, a cranial cardinal sinus that empties into a common cardinal vein, which then goes back to the sinus venosus. Here you can see the arteries of a shark from the gill region. These are those efferent arteries bringing oxygenated blood from the gills, emptying into a dorsal aorta, but this then would include the internal carotid arteries, which would go to the brain and service those. All right, and so here, once again, you can see the efferent arterioles joining to the dorsal aorta. Once you distribute red oxygenated blood throughout the body, gas exchange occurs in the tissues. You now have blue deoxygenated blood, which then returns as I had mentioned. Okay. So you would see about the same setup in bony fish. But getting to the Sarcopterygian fish, there is an incredibly important develop, obviously. Once again, those basic 6 arches can vary, and there is a reduction in these over time. Once again, we have an internal carotid and an external carotid here. The key is Sarcopterygian fish have a lung, like lungfish, hence their name. And the lungfish, they will service the lung with a branch of the 6th aortic arch. So these are numbered, all right? And the numbers are important because some of them are lost, like the 1st aortic arch. Some will be kept, as we will see in mammals. One of the ones which is kept and important is the 6th aortic arch. So what can happen is a lungfish can send deoxygenated blood to its gill arch, and then the blood becomes oxygenated and it can now get sent throughout the body. But what happens if the lungfish is in stagnant water and the gills aren't getting enough oxygen? So the oxygen coming from here just isn't enough. Well, now relatively deoxygenated blood can then be sent to the lungs. So gas exchange can occur here from the 6th aortic arch, but not from the gills, or not entirely by the gills, but now in the lungs. From the lungs, then a vein, a pulmonary vein, goes into the left side of the heart, while humans have right and left atria. and the left atria receives the pulmonary veins. Here we can see the beginnings of that, where a pulmonary vein empties into the left side of the atrium with a partial interatrial septum. So we see the beginnings of the separation of oxygenated and deoxygenated blood that we will see later in the mammals. Notice there's still a connection between this sixth arch and the dorsal aorta, known as the ductus arteriosus. When we humans had were embryos, we still had a ductus arteriosus, which connected the pulmonary trunk to the aorta. So we still had this, but it is lost, but it is retained in adult lungfish. It will also be retained in adult frogs. And so if we were to now consider what does a frog's circulatory system look like. Once again, starting off with those basic aortic arches, we have an internal carotid and external carotid. There are lungs in the frog, but now notice we've reduced the aortic arches to three. The third aortic arch, the fourth, and the sixth. The third then becomes the carotid arch, which services the head. And so we have going from the heart, there is still the aortic arches going here, and external carotid will come. Interestingly, there's this little body here called a carotid body, and here there are some sensors which help detect blood pressure, the oxygenation of the blood, and we humans, we still have, although we don't have a swelling like exists in frogs here, we do have some of the sensors which are detecting the chemicals in our blood, like carbon dioxide, oxygen, to determine how our respiratory system needs to adapt. We still have it where the common carotid branches to make the external and the internal carotid. So this carotid system is then a modification of the third aortic arch. And the common carotid, which did not exist in fish, then becomes a blood vessel in the land of vertebrates. And we humans, coming off the arch of the aorta, we have a common carotid artery, which then branches to the external carotid artery to the face and the internal carotid artery to the brain. And once again, we have those special sensors right here. So that's what happens not only in frogs, but in the third arch of mammals. So when we were embryos, we had a third aortic arch that then becomes the carotid arch, giving rise to the common carotid, external and internal. The fourth aortic arch is important because this then becomes our aorta. All right, and so in frogs and humans, there is an aortic arch, which then forms the dorsal aorta. There may be one dorsal aorta or two. Remember that these are paired at first. And so you can see on a reptile, there's both aortic arches. Some animals only keep the right one. We humans only keep the left aortic arch. But getting back to frogs, obviously after the tadpole stage, there are no gills. And so then this sixth aortic arch then becomes special because it then is primarily now traveling to the lungs. And so the frogs, they separate their deoxygenated and oxygenated blood into a left and right atrium. They have one single ventricle, but nevertheless, there is, even you would say, oh, that seems inefficient. The blood all mixes here. Yeah, it can mix to a degree, but between these pumping not at the same time or pumping getting fluid in different directions. So in a heart, even in humans as fetuses, if you have blood all in the same chamber, if it's coming from chambers at different speeds, at different angles, you can still largely separate two pools of blood in one single chamber. So the blue deoxygenated blood is staying in this sixth arch, which then becomes the pulmonary loop. We humans, our pulmonary circuit, right, where we have pulmonary arteries which go to the lungs, is a modification of the sixth aortic arch that we develop in embryos. Frogs still have this ductus arteriosus, which isn't arguably the best anatomical setup because it could potentially bring deoxygenated blood into now the red oxygenated blood. Here, we humans, we don't have that as an adult, just as an embryo. Okay, so if we were to actually then look at the frog blood vessels and trace this flow. So once again, deoxygenated blood is going to come in through the sinus venosus. Now there are two atria. There's a separation between the right atrium receiving deoxygenated blood from the sinus venosus and the oxygenated blood from the lungs coming into the left atrium. So deoxygenated blood into the right atrium, into the single ventricle, and then out the truncus arteriosus. But from there, where would it go? Well, this deoxygenated blood needs to be oxygenated. And so there is coming off the arch, there are now arches derived from these gill arches. And so the third arch, once again, is going to the carotid region. The fourth is the dorsal aorta, and then the sixth is going to the lungs. And so here you can see the carotid arch. That's arch number 3. So in fish, that would go to a gill, but now it's forming a common carotid. There's the carotid bulb, and then an internal and external carotid. Then you see the fourth arch that then forms the systemic aorta, the systemic arch, and then you see a sixth arch going to the lungs. In frogs, this is called the pulmonocutaneous artery, because in addition going to the lung, here's the pulmonary artery, it also goes to the skin, and that's a route that frogs can get a lot of blood from as well. So if we were going to trace blood flow here, I'm going to use blue and red colors, but I just make a note because there can be some mixing in that one single ventricle. Just, the colors aren't quite as accurate as they would be, if I were describing, say, human circulation. So we can go from the sinus venosus to the right atrium, to the ventricle, to the conus arteriosus, to the pulmonocutaneous artery, to the pulmonary artery, to the lung. Once again, this is that sixth arch now. There we have gas exchange in the pulmonary capillaries. A pulmonary vein goes into the left atrium, something that no fish has, a completely separate left atrium. The oxygenated blood then goes into the single ventricle again, then into the conus arteriosus. And if we were going to send it to the brain and back, we would then go into that third arch, the common carotid. Will then go either to the external carotid to the face, or because we're talking about the brain and back, to the internal carotid, which would go to the brain. Their gas exchange occurs in the capillaries of the brain, and now deoxygenated blood would return through a jugular vein and into an anterior vena cava. There would be 2 of those into the sinus venosus. Now, that's how you would get oxygenated blood to the brain if the oxygen was coming from the lungs. Frog lungs don't work very well. And so frogs also do an awful lot of gas exchange with their skin. So here you can see the skin of a frog. Look at all those big blood vessels. Why so many blood vessels in the skin? Because that's where a lot of gas exchange is occurring. And that's why frogs need to keep their skin moist. And so We have the pulmonocutaneous artery has one branch, the pulmonary artery going to the lung, but then the cutaneous arteries then go to the skin, and a great deal of gas exchange can occur there as well. And so once again, amphibians have lungs. Doesn't work they don't work as great as the lungs will in the amniotes. And so the skin, which now must be kept moist, then supplements that. So frogs are getting oxygen from two sources, their skin and their lungs. But interestingly, both are coming from that sixth arch, which gives rise to both the pulmonary artery and the cutaneous artery. Just identifying some of these blood vessels in the frog, you can see that there is a common carotid. That was that third aortic arch. And then right where the branch occurs, you see the swollen area, the carotid bulb, where those sensors, which are sensing how much carbon dioxide is there in the blood, we have the same. This is right where the common carotid will split to make an internal and external carotid. Once again, where our common carotids, we humans, where it branches to form the internal and external. We also have those special sensors. So this then is how brain blood gets to the brain, and this is that modified third aortic arch. And then once again, if humans are trying to control their respiration and determine, do we need to breathe more deeply, less deeply, etc. Where are we perceiving this? Where the medulla can perceive a little bit of what's our oxygen, acid, carbon dioxide levels. So can our aorta. But we also have carotid body sensors in the common carotid where it branches. So salamanders vary a bit, and obviously salamanders, some larvae spend lives in the water and have external gills, as some larvae just kind of develop in the egg stage and hatch as miniature adults. Some adults remain aquatic and then are breathing through external gills throughout their entire lives. And so, obviously salamanders vary a great deal. And so the circulatory routes can vary from one salamander to another, especially how the larval circulation is modified to make the adult circulation. But if we, take this salamander, we bring deoxygenated blood from the sinus venosus to the right atrium. Once again, amphibians have separate atria and ventricles, separate atria, into the one single ventricle, into the conus arteriosus, to the pulmocutaneous artery. Once again, it services both the lungs and the skin. From there, the pulmonary artery can go into these elongated lungs. Salamander lungs are very long. This then comes into a pulmonary vein into the left atrium. Once again, salamanders have separate left and right atria, into the ventricles, into the conus arteriosus, into a common carotid, to the internal carotid, to the brain, and then after gas exchange occurs in the brain capillaries, back through the internal jugular here. Now, once again, there are variations in salamanders, as I mentioned. Here you can see one as an adult is remaining aquatic and therefore still retains those external gills. The most common salamanders in the state, or the Northeast United States, in the state of New York, Pennsylvania, is the family Plethodontidae. They have no lungs. They are lungless salamanders. And so here you can see, while many salamanders have these long elongated lungs, and here you can see the pulmonary veins bringing oxygenated blood back from the lungs. Many salamanders, the most common family in the Northeast US, the Plethodontids, they lack lungs and are doing all of their gas exchange in their skin. There is one frog which also lacks lungs. And so therefore that the pulmonocutaneous artery wouldn't have a pulmonary branch. Some other factors as well. In lung lists, salamanders, the interatrial septum and spiral valve and the conus arteriosus may be reduced or absent. So once again, groups don't stop evolving as we see, and there can be some variations throughout different groups. Okay. And so once again, I'll make a big deal about splitting the ventricles in the reptiles, which have a partial interventricular septum, and in the birds and mammals, which have a complete interventricular septum. But there is actually a group of salamanders, the sirens, which have a partial interventricular septum, which is more advanced than the situation you would find in most salamanders. Just a quick note on the salamanders, the legless salamanders, they can actually, while they're performing their gas exchange through the skin, it is not the pulmonocutaneous artery which is doing that. That is completely absent. And now it's just separate arteries coming from systemic circulation from different parts of the body. Sorry. So let's now talk about reptiles, and we'll use a turtle as an example. Now, obviously, we don't have the very first reptiles, alive today to dissect, and so therefore, we pick a reptile. Each one would be differences between alligators versus lizards versus turtles. But let's just pick the turtle. So here you can see the right atrium and the ventricle. Reptiles have separate left and right atria, but they have only a partial division between their ventricles, so it's not complete. And so here you can see the atria, the ventricle, you can see the pulmonary trunk, which is going to the lungs, and then you can see the aortic arches, which will be the systemic arches. Remember, that's the fourth aortic arch, which is going to service the systems of the body. There's actually two halves, because the gills are on both sides. And so while we mammals, we have the option of having two aortas, one deriving from the fourth arch on this side, one derived from the fourth arch on this side. We just keep the left. I'm sorry, it's my right hand left in the screen, sorry. We then, turtles actually keep both. So there's a left and a right half of the aortic arch. But because I'm talking about the brain and back, we'll be focusing on the third aortic arch, which will then give rise to the carotids, which go to the brain, but also the subclavians, which go to the arm. Right. So once again, just getting back into turtles, you can see that there's a pulmonary trunk derived from the sixth arch. This then gives rise to so if I follow it here, to the pulmonary artery, which will go to the lung. Here's the lung here. That then comes back through a pulmonary vein into the left atrium. So just like we have a pulmonary artery, goes to the lung, comes back a pulmonary vein, the left atrium, same thing here. So turtles still have a small sinus venosus that empties into the right atrium, the ventricle, which only is partially divided. Then from the pulmonary trunk to the pulmonary arteries to the lungs, gas exchange occurs. We now have red oxygenated blood, which comes into the pulmonary vein to the left atrium, the ventricle, and then goes into the ascending aorta, which then goes into brachiocephalic veins. There are brachiocephalic arteries. There are two of those, as opposed to us, which just have one. And after the subclavian branches off, then the common carotid will then take the oxygenated blood to the brain, where the internal carotid reaches the brain, and then the jugular vein will bring it back. to the sinus venosus. And so here you can see there is a common carotid artery after the subclavian branches off to go to the arm. This was a brachiocephalic trunk earlier. And notice how long it is, because in amniotes, the neck is long. Amphibians have a very short neck. Amniotes lengthen their neck, so the common carotid gets longer. Once again, the blue blood in the sinus venosus comes into the right atrium, into the ventricle, and from there we'll go into that derivation of the sixth aortic arch, that pulmonary arch. So here's the pulmonary trunk here. From there, there will be a pulmonary artery which reaches the lungs, which are here. Gas exchange occurs. A pulmonary vein will come back into the left atrium, which is here, and then from there, this red oxygenated blood goes to the ventricle, and it will then go into the brachiocephalic trunks. There are two of those. Once again, we humans, we only have one brachiocephalic trunk on the right side, and then from the brachiocephalic trunk go into the common carotid, which will then carry blood to the brain. Okay. Birds have about the same setup. The one big change in birds is that there is a complete septum between the left and right ventricles. So birds have separate ventricles, just as mammals do. So the deoxygenated blood is coming back from the brain through from the jugular veins into the precava, or the anterior vena cava, into the sinus venosus, into the right atrium, into the right ventricle, which is separate from the left ventricle. Once again, there's a complete interventricular septum. You then go from a pulmonary trunk to pulmonary arteries. The lungs perform gas exchange. Oxygenated blood comes back through the pulmonary veins. And then from the left ventricle, once again, that is separate from the right ventricle, blood goes into a descending aorta. Now, once again, the aorta is derived from the sixth arch, and we have two gill arches too. And so we could have both arches kept, as turtles do. We mammals, our aortic arch, we only keep the one on the left. The one on the right degenerates. Birds do the opposite. They keep the one on the right. So their descending aorta is only their right aortic arch. But since we're following blood to the brain and back, the brachiocephalic arteries go to the common carotid artery going to the brain, and the subclavian going to the wing. And so then the common carotid will reach the brain, and we'll have gas exchange after the internal carotid reaches brain capillaries, etc. Okay, so fairly similar to what we would see in humans. So once again, there are two brachiocephalic arteries. There's a common carotid, internal carotid, and then blood would come back through the jugular into the precava. Okay. Then finally, we could consider then how mammals get blood to the brain and back. And once again, as we look at this ancestral pattern, which we develop as embryos, we modify these aortic arches, and so that we're only keeping the third arch. This is the carotid arch. We keep the fourth arch. This becomes the systemic arch. And then we keep the sixth arch, becomes the pulmonary arch. The ductus arteriosus exists, connecting the fourth arch and the sixth arch as embryos. But then when we're born, this is lost, and we just have a ligament here. The ligamentum arteriosum replaces the ductus arteriosus, which we had as embryos. So once again, because this could develop on each side, we could have two fourth arches forming two dorsal aortas, but we only keep the one on the left. So we only keep the left fourth systemic arch, and mammals once again have a complete ventricular septum separating their left and right ventricles. So this systemic circuit, you can see this, I'm sorry, this pulmonary circuit, you see the pulmonary circuit is completely separate now from the systemic circuit. There is no connection between the two the way there was ancestrally. Now, I have videos which trace the blood flow in humans going to the brain and back. A lot of anatomy students obviously want to go into the study of human anatomy, but here just ending with the opossum and the cat. One difference we see in the cat is that we see there are two superior vena cava emptying into a common area, which I think is the remains of the sinus venosus. Placental mammals don't have this, but obviously they're entering into something before they enter into the atrium. So I haven't read this, but it looks like a sinus venosus to me. And so here you see there are two superior vena cava, whereas humans and placental mammals just have one typically. The right atrium is collecting blood from the superior and inferior vena cava, and also the coronary. I'm sorry, there's the sinus venosus coming in. And so, because we mammals, we have a complete interventricular septum. Notice that's complete. So the right ventricle and its deoxygenated blood is completely separate from the left ventricle and its oxygenated blood. And so from here, the arch of the aorta, which once again, we only keep the left arch, then forms a brachiocephalic trunk, which then forms common carotid arteries. This is a little different from what we would see in a human because the left common carotid does not come off the brachiocephalic trunk. It comes off of the arch of the aorta. So here's an arch of the aorta with one brachiocephalic trunk that's giving rise to all of the blood flow to the brain. In humans, the blood flow to the left side of the brain is not coming off the brachiocephalic trunk. So, some minor differences here. Notice that the left subclavian artery and the left common carotid, once again, are coming off that brachiocephalic trunk instead of off the arch of the aorta as in humans. Here you can see the jugulars coming back from the brain. And once again, notice that there is a pair of superior vena cava and one inferior vena cava in the opossum, making it a little different from what we see in the placental mammals. If you're looking here then at a cat, it's mostly the same as what we would see in humans. Notice that there is one brachiocephalic trunk on the right side, and the left subclavian is coming off the arch of the aorta. One small difference than in humans is that in humans, in addition to the left subclavian coming off the arch of the aorta, so too does the left common carotid come off the arch of the aorta. Here in the cat, you'll notice both common carotids are coming off of the brachiocephalic trunk. And so once again, there's the brachiocephalic trunk. There are the two common carotids, both coming off the brachiocephalic trunk, which is unlike the situation in humans where the left common carotid comes off the arch of the aorta. Okay, so this has been an introduction into just blood vessels in general, and then how vertebrates get blood to the brain and back. I have more specific human videos elsewhere, because obviously in humans there are two routes in addition to the common carotid. You can also get blood through the vertebral artery as well. And my next video will then consider blood flow to the limbs.